Our next illustrious speaker is Fernando Rivadavia. He started growing plants and being interested with them again when he was a little boy, when he was 13. He doesn't grow any plants right now, I don't think. But he is an expert on these plants. He has described 26 new species of carnivorous plants, sundews, Utricularia, Gillissia, even an important one, even a helianthera. Yeah, lots of awkward selfies with plants all over the world. At any rate, I'd like to give you your friend, Fernando Rivadavia.
Okay. Thank you, David. Good morning, everybody. Thank you for being here early on Sunday. Hopefully, everybody got some sleep. Hopefully, you got some good, strong coffee, because I'm going to try to make this talk not very technical. I hope there's a little bit in it for everybody. I'm going to discuss carnivorous plants phylogeny, so basically the evolution, what we understand, it's a summary of what we know of carnivorous plant evolution in terms of how they're related to each other right now. I'm going to summarize a lot of the work that's been done up to now and where I think things are going to go in the next few years. So in order to be able to do that, I'm going to give a little brief introduction. What is DNA? What is DNA sequencing? Just to remind some of you who's maybe it's been a while since you've seen it, maybe you never saw it, and things are changing very fast. What we know of DNA is evolving very fast. What I learned about it 20 years ago in college has changed a lot. And then I'm going to go into a little bit what is a phylogeny, what is a molecular phylogeny, that is phylogeny using DNA instead of morphological characters, for example. Very briefly, just mention the names of the genes that are mostly used for phylogenies, just because I'm going to be saying them over and over again. And then we're going to dive into the different families, different genera. And like I said, I'm going to summarize a little bit of what we know currently, how they're related or not. And finally, at the end, talk about where it's going and what we should expect to see maybe by the next ICPS conference in 2020. Maybe by 2030, what we should be seeing at the conference, what sorts of talks we'll be seeing, what sorts of plants we'll be seeing in the show. And then again, 2040. So where are things moving? So first of all, DNA, probably most of you know the double helix. DNA are two strands of A's, T's, C's, and G's. So chemical letters, and this is a code. So all animals, all plants, viruses, bacteria, everything that's living has DNA, or sometimes its partner RNA. So strings of A's, C's, T's, and G's. But before we get a little bit more into that, where is that all located? So looking at a plant cell, there's a lot of stuff in there. We're going to focus just on a few little things. The nucleus, the chloroplast, and the mitochondria. Nucleus is where most of the DNA is located in plant cells. But there's these two little organelles here, chloroplasts and mitochondria. We have mitochondria as well, but we do not have chloroplasts. Mitochondria is the powerhouse of the cell. So, yeah, it's present in animals, plants, fungi. And chloroplasts are the ones that make photosynthesis. So you have them in plants. The curious thing, though, is that both chloroplasts and mitochondria have their own DNA. It's only a little bit of DNA. It's a little circular molecule instead of a long chromosome like in the nucleus. But they have their own DNA. And this is transmitted from mother to child. Okay, both chloroplasts and mitochondria, in general, most plants and in animals as well, are transmitted from the mother to the child. We do not get it from our fathers. So it shows a maternal lineage, maternal inheritance. And just a little curiosity, some of you may know, these two were originally bacteria that got incorporated into animal and plant cells. That's why they have their own DNA still. Okay, so we are actually mutualistic organisms that fused together millions of years ago into what is we call a eukaryotic cell. Okay, so in the nucleus is where most of the DNA is. You got one nucleus per cell usually. There's lots of exceptions, but the DNA is usually contained in large chromosomes, so strings of A's, C's, T's, and G's. Usually you get half of your DNA from your mom, half from your dad, except for the chromosome mitochondria, which we'll go back into. And you usually have two copies of each gene, so two alleles. So you have one allele on the paternal chromosome, one allele on the maternal chromosome. Sometimes they're the exact same sequence. Sometimes slightly different sequences, so that one may code for the classic blue eye versus green eyes or some other character. But sometimes we do have genes that are present in multiple copies. So you don't have just one copy on your maternal chromosome and one on your paternal chromosome. Sometimes you have 5 copies, 50 copies, 500 copies. And one we're going to be talking a lot about is one called 18S, something called ribosomal DNA. Animals and plants tend to have dozens of copies, and so it's very useful for phylogenies because it's easy to pick out of the crowd, let's say, and amplify and sequence. And plants have an added complication. As a lot of you growers may know, plants sometimes double their chromosome content. And let's say they originally had 20 chromosomes, sometimes they'll double and have 40 chromosomes. So 2 sets of the whole chromosome, of the whole genome. Sometimes they're doubled, sometimes tripled, sometimes 8, 12, or more. And that makes sequencing sometimes a little more challenging because you don't know exactly which chromosome copy you're sequencing, whether they are actually the same sequences or not, whether it's a recent polyploidization event or an old polyploidization event. So plants have an added level there that you don't usually see as frequently in animals. So remember, nucleus is only 1 per cell usually. Sometimes many copies of each gene, but mitochondria and chloroplasts usually have dozens, hundreds of copies per cell. So a lot of times scientists like to use the genes in mitochondria and chloroplasts for phylogenies because if you extract DNA from a tissue, you'll have maybe millions of copies of these guys and maybe a few hundred copies of the nuclear genes. So it's basically easier to work with. So sort of the lazy way to do phylogenies sometimes is use genes that are easier to pick up from the crowd. As I was saying, it's maternally inherited, usually. So usually when we look at phylogenies, at those phylogenetic trees made from chloroplast genes or from mitochondrial genes, we're looking at maternal lineage. And that's very important with plants, because as you know, there's a lot of hybridization in plants. Plants don't, more than animals, evolve in a reticulate manner, where 2 species will come together and form a new species. Sometimes you'll get more than 2 species. For example, wheat is a hybrid of 3 species, the wheat that we normally consume. And who knows what else is in there that we haven't found out yet. I mean, there's tons of plants that are probably hybrids. In fact, probably everything is a hybrid. Every single plant, if you go far enough in its background, in its ancestry, you'll find hybrids. So that's going to complicate things a little bit further on, as we'll see. Okay, and as I was saying before, mitochondria and chloroplasts, so they do have their own DNA. It's a tiny little round molecule, chromosome, just like bacteria have it. Back to DNA, so most of the DNA is in the nucleus of the cell. Part of it is in the chloroplast. In the mitochondria, we call that the genome. Right? So the genome is a complete set of DNA that a cell has. And every cell usually has the same set of DNA throughout the body, with exceptions. And what is the DNA? It's a code to make proteins. Our bodies don't work without proteins, especially like digestive enzymes, structural proteins. DNA is there it's a code to tell our bodies how they should build themselves when they're developing in the womb. It's how we should react to different environmental conditions. So DNA is the code. It gets translated into what we call RNA molecules, and then that gets translated into proteins. That's a central dogma of biology. Okay, so DNA to RNA to proteins. We're not going to worry about RNAs. We're not going to worry about proteins in this talk. We're just going to focus on the sequence, the core of that code of the DNA. DNA sequencing started back in the 70s with a method called Sanger sequencing. It was initially tremendously laborious. To get a single sequence like that might take you a week or more. Initially, the sequences, the largest that we could get were maybe 200, 300, 400 bases at a time. So small snippets of genes. Right? If you remember, the human genome is 3.2 gigabases. That is billion A's, T's, and G's. So here we're sequencing just a tiny little snippet of maybe a single gene, and then we're going to compare it with other sequences and try to see how closely related species are. So the Sanger sequencing started in 1977. Initially, it was, like I said, tremendously laborious. Took tons of time, tons of work to get small sequences, but that changed. So it got faster and faster. We were able to sequence the first human genome back in 2003. It was completed. at a price of $3 billion. Hundreds of labs, thousands of scientists throughout the world sequenced the first human genome. For those of you who are alive, you may remember it was a huge feat. It was tremendous. $3 billion for one genome, 15 years. Well, that has dropped. Over the years, machines have gotten faster. We've got now what we call next generation sequencing, using new chemistries, new instrumentation that sequences in parallel, tons of stuff at a time, and the cost of one human genome has dropped significantly over the years. We are now at a place where we can sequence a whole human genome for less than $1,000. And when I say we, I say one single person can prepare the samples in the morning, can load it on an instrument at night, have the results by the morning. So what took thousands of scientists 15 years ago, thousands of scientists, $3 billion, a single person can do it now on the largest sequencing machines. So what does that mean? That means that as DNA sequencing gets cheaper and cheaper, it allows us to do all these things that were previously impossible because it was just too expensive. So it's given us tremendous power to look further and further into the genomes of these plants. And as you'll see, a lot of the early phylogeny started with a single gene. Oh my gosh, I got a single gene sequence for maybe 5 plants. That was amazing. Now we're sequencing whole genomes of plants and comparing whole genomes at a time. But I'll get there. I'm jumping a little bit ahead. So let's talk phylogenies. What are phylogenies? Usually this is what you'll see in a phylogeny, a phylogenetic tree. And I put this one here because I'm biased. This is my phylogenetic tree. For my little plants, my sundews, of course. But here's an example. How does it start? So you need to sequence a snippet of DNA. And you're going to look for the same snippet in all the different plants you want to compare. So you choose a gene, or a gene. I'm not going to go into the discussion of what is a gene, because it's as contentious as the discussion of what is a species. So just consider gene any segment of DNA. So I choose a specific gene that codes for, I don't know, leaf length or whatever character, it doesn't matter. I sequence that gene and I compare the A, C's, T's, and G's across that gene for several species, in this case Utricularia. And what I'm looking for are things called SNPs and indels. SNPs are when you notice at one position, hey, look, all these species have a G, but hey, you rigid, that has an A in that position. Ah, that makes you rigid a different. If I sequence more species later and see that there's another 5 species that have an A, that tells me that maybe the ones that have an A are more closely related than the ones that have a G. So I have 2 groups, the G's and the A's in that specific position. I'm also looking for indels. Sometimes little snippets of DNA get inserted or deleted. Which one happened here? I don't know. But this guy has an insertion relative to the other guys, and these guys have a deletion relative to this guy. That's an important character there. That's going to help me build a tree as well. Say, hey, this guy's a little bit different. And maybe I'll find other species. For example, Juncia, we know it's closely related to Cornuta. If I sequence Cornuta, very likely I'll see that little insertion there as well. I'll say, these 2 are more closely related than all the others. It's like when you're comparing morphological characters. You see 2 species that have especially odd character, I don't know, really long leaf or red flower or whatever, we'll say, oh, maybe they're more closely related. But sometimes not. Sometimes it's convergence. Sometimes 2 different species have a similar character, like, I don't know, classical bats and birds that have wings and think, oh, they're closely related. No, completely different convergent evolution. And sometimes here, also, things will happen where you'll get a mutation, but another plant will get the same mutation. And you'll think they have a common origin, but they don't. So you often have to sequence more and more and more to weed out some of these little errors. Yes?
What's the Q in the second line? Just
There's a Q in there. Damn it, I thought I'd weeded those out. A Q, there's a lot of other little letter codes that mean that there's already a known SNP in there. It could be one or the other. I don't see where the Q is, but...
second line down, just got right of the gap.
Right of the gap. Q. Oh, it's a small g. Sorry. Somebody just... usually they'll put a lowercase a, t, c, or g when they're not sure. That electropharogram before, where was it? Was not too clear. Maybe the peak was a little low. So they're like, I'm going to put a little g until I can sequence a little better and be more certain of that sequence. That's just a little trick that phylogenists often use to tell themselves that, you know, I got to take a better look at this, see if it's not something else afterwards. Okay, so in order to build phylogenies, we choose a gene or multiple genes. We sequence those across many species, maybe many genera, many families that we're comparing. Then we look for SNPs, single nucleotide polymorphism, little mutations, or we look for insertions and deletions. And there's a few other characters I'm not going to go into, but that's what we're looking for in order to build phylogenetic trees. So based on how similar the sequences are, or how different they are across the species that you're looking at, you can build little phylogenetic trees. And they can look anything like, they can be on the side, they can be facing up, they can be, more square, more triangular, whatever you want to call that. Basically, we're seeing that A and B are closer to each other than they are to the other species. C and D are closer together, more closely related than they are to the other species. And E is sort of like a basal branch. It doesn't mean that E is more primitive. It just means that it diverged, it separated more millions of years ago or something, okay? But this guy has as long an evolutionary history as all the other ones. So this guy might look a little bit more like the ancient ancestor, or this guy might look completely different from the ancient ancestor, and this guy might look more like the ancient ancestor. So never assume that whoever's at the base is more primitive and looks more like the original ancestor. Okay, so these could be different species. They could be different individuals in a population. They could be different genera of carnivorous plants, could be different families. You choose phylogeny. Sometimes people are looking at a bigger picture, family. Sometimes people are looking at a small picture, individuals in a population. Either one. And we say that they're monophyletic when they're all on the same branch. So these are all monophyletic. Okay? But sometimes what we consider monophyletic, when we actually sequence and we say, hey, they're paraphyletic, that is, sometimes A and E are classed together in, they're Drosera, and then somebody says, oh, but wait, see, here I have Venus flytrap, Eldravanda, and Drosophilum. So wait, if that was true, then this would be completely paraphyletic. Luckily, that doesn't happen too often with our carnivorous plants, but so the important thing is that a lot of times in traditional taxonomies, what we used to lump together when we sequence, we say, hey, remember the bats and the bird? They have wings. We thought they were close related. Actually, a lot of times they're not. So with molecular phylogenies, because we have so many characters to work with sometimes, it helps us identify possible convergences of where we're lumping things that shouldn't actually be together. At least that's the hope. Because sometimes we sequence things and we see what we call polytomies. We're like, crap, look, they're all branching together. It seems like they all diverged at the same time. And sometimes it just means that you didn't have enough sequence in there. You didn't have enough SNPs and indels to use as characters. And basically, you're going to have to sequence more in order to find out how they're related. So polytomies are very common. I'm going to show you a lot of different phylogenetic trees from carnivorous plants. You're going to see polytomies all over the place, unfortunately. So what is the secret sauce of phylogenies? How do you get a good phylogeny with more dichotomies and less polytomies? You got to find the right gene for the right group of plants that you're working with. So you got different pieces, different parts of DNA evolve faster or slower within a genome. Some genes are under tremendous pressure not to change because they're extremely important to how our bodies work. So if you compare one of these highly conserved genes between several closely related species, you may not see a single SNP, a single mutation. They might have the exact same sequence. Whereas if I sequence a part of a gene that there's little snippets of genes that are not exactly translated into proteins. And these snippets of what we call introns or spacers, they're more flexible. They can incorporate some mutations, and it doesn't really affect the protein that they're coding for. So these tend to evolve a little bit faster. So if I'm looking at plants that are very closely related, usually I'll go for these genes that are very fast evolving. Whereas if I'm looking at a higher level at families, comparing the Droseraceae with the Nepenthaceae with the Triphyophyllum, maybe if I go a little bit higher and grab a gene that doesn't evolve so fast, will work better. And that's really hard because when you start, you often don't know. Because what is a fast evolving gene in one plant family may be a slow evolving gene in another family. So often what phylogenists have to do, they have to sequence several genes and compare maybe 2 closely related species and 2 more distant related species within the group that they want to study. They need to do that first before they can actually dive in and sequence everybody. So this can be tough, and a lot of times people get it wrong, as you'll see in some of the phylogenies that are coming up. So let's talk about genes. So genes, not those genes, of course. We're talking about, like I said, the snippets of DNA. Throughout this talk, you guys are going to see this one come up over and over and over again. Nuclear ribosomal DNA. It's one of those genes that, like I said before, is sort of a lazy gene because there are sometimes hundreds of copies, thousands of copies within each cell, within the genome, the nucleus of each cell. Why exactly? Because it just produces a protein that's very important to our bodies that we need a lot of. So we need a lot of copies in order to make a lot of that protein. Now those copies, often they'll be the same sequence, often they'll have different sequences, and we never really know until we sequence it. And sometimes one species will have two sequences and only one will pass to its descendants, or only the other will pass to its descendants. So sometimes we're comparing things that we think are all the same and they're not. Because again, the reticulated evolution in plants messes things up. So we're going to see things like 18S, we're going to see ITS coming up often. It's a more fast evolving spacer area, an intron. So just so you know, this is going to come up over and over again. It's a nuclear gene, that is, it's in the nucleus, and we not only have two copies, one from the mom, one from dad, we have tens to tens of thousands of copies. And you're also going to see a lot of chloroplast genes showing up. So you're going to see little things like TRNK, MATK, RBCL, a whole bunch of chloroplast genes, which means that they're maternally inherited. So when you hear 18S and any of its components, you're thinking, okay, that comes from the mom and the dad. It's showing not necessarily just a maternal lineage, it's showing maybe a more complete view of the evolution, whereas this one can be extremely biased because it only shows maternal lineage, which, if it's in a genus like Sarracenia that has a lot of hybridization, may not show the full picture. But let's skip that. Okay, so you don't need to worry about the names of genes. Just keep in mind that they're nuclear genes, come from the mom and the dad, and chloroplast genes, mitochondrial genes that come from the mom only. Okay, let's dive into phylogenies. Let's start. The first carnivorous plant phylogeny was published back in 1992. It's old already, but I remember when it came out, it was huge. This was done with the old Sanger sequencing method, a manual method that was tremendously laborious. I know one of the authors, Victor Albert, and man, he was telling me about how much work it was. He had to make these huge gels and put this photographic film on top and leave it for days, and then the gel would break when you pulled it apart. It was awful. But what did this phylogeny show us? What a few speakers have talked about already. First, that carnivorous plants are not monophyletic. That is, they don't have a single origin. They have multiple origins. Carnivorous plants didn't only evolve once. They evolved multiple times, maybe 5, 6. And who knows, maybe there's been other carnivorous plants that we've lost that have evolved in the past that we don't even know about. So what do we see? Well, what we expected, for example, Pinguicula, Utricularia. Gillisia is not shown there, but afterwards it was shown, yep, they're monophyletic. They have a single origin. So there we have a family with 3 trap types in a single family. Pinguicula, sticky trap. Gillisia, it's almost more like a pitcher plant if you think of how Gillisia works. And then we have Utricularia, it's a bladder, an active bladder that sucks in its prey. So 3 completely different trap types in a single family. And then they also included a few sundus. So we have there Drossobinata, Peltata, Pygmies, etc. We also have Nepenthes right over here. We have Drosophilum, Dionea, Venus flytrap over here. Afterwards, Aldrovanda was shown to be in there as well. Triphophyllum was shown to be close. So even though it's not one family, they do seem to have a common origin there. Triphophyllum, Nepenthes, Aldrovanda, Dionea, Drosera, all seem to have a common carnivorous plant origin. And that's going to be very interesting a little bit further on. They did a nice little selection of sundus there. I don't know if this is accidental or not, but they chose like some of the most basal ones, some of the most derived ones. What else do we see? Darlingtonia, Sarracenia, Helianthera. It was a bit of a surprise to see Darlingtonia at the base of the genus. Everybody always assumed that Helianthera was a more primitive one. It had sort of like the more simple traps, and it has the more different flowers than the other two. So that was a little bit surprising. Again, it doesn't mean that primitives, the ancestor of these guys, looked more like a Darlingtonia. It could be that they did look more like Helianthera, but Darlingtonia has evolved a completely different trap. Remember, it's just as old. These 3 are just as old. But for Darlington is a little bit older, branched a little bit early. It's been evolving on its own for a little while longer, according to this tree. Oh, and before I forget, every single slide where I have a phylogenetic tree, I'm going to have a list showing which gene was used. Okay, in red. RBCL, that's the chloroplast gene. So this is maternal lineage. So this is all based on a single gene from a chloroplast maternal lineage. This may all be wrong. Who knows? Once we sequence more information, we may see that Helianthra really is at the base. We don't know. This is preliminary data from over 20 years ago, almost 30. Geez. So let's take things slowly, one at a time. We'll see more phylogenies with more genes, how it changes, okay? But the really surprising thing here was this guy, Rorigula. Who would have thought that Rorigula was closely related to the Sarracenia? All right, this is a complete surprise. So interesting, here we have two very closely related carnivorous plant groups, one with a sticky trap, one with a pitcher trap. Interesting, we've seen that before over there, right? And we've seen it here. Nepenthes close to sticky traps as well. Interesting, snap traps close to sticky traps. And then we had old Cephalotus showing up completely somewhere else on the tree. It's all on its own. There's no other carnivorous plant that we know of that is closely related to Cephalotus. Who knows how it originated, what the original Cephalotus ancestors looked like. We have no clue. But one very good example is that little group that I'll show you, Nepenthes, Triphyphilum, Drosophilum, Aldrovanda, Dianea, and Drosera. It seems like the ancestor was a sticky trap. So there was some pre-adaptation. I'm sure many of you know plants that are glandular, that trap insects, apparently accidentally. A lot of them have glands on the sepals around the flowers, maybe to keep ants from climbing up and stealing the pollen or stealing the nectar. So it may have started just as a defensive mechanism. And if you think of a sepal, it's just a leaf, and the tentacles then would be on the back side of the leaf, right? Have you ever looked at Drosophilum? The tentacles are on the back side of the leaf. And then on Drosera, it flips to the upper side. So apparently, in fact, one of the plants in this group is Plumbagua. I don't know if you guys know Plumbagua. Yeah, it has a glandular hairs on the sepals, traps a lot of bugs, probably protecting the plant. So at some point, maybe those glands started digesting the bugs. There's digestive enzymes in the seeds that are normally present in seeds that might have been accidentally expressed in the sepals. And hey, all of a sudden they're digesting prey. Maybe they're getting a little bit of nutrients, getting an advantage over their neighbors. So apparently the traps started in this group as sticky traps. Tentacles elongated, started moving. Maybe the leaf started moving as a whole. In the Drosophilum and Triphyphilum, the glands don't move, but they are extremely sticky. And then some of these flypaper traps evolved into snap traps, where some of them evolved into pitcher traps. So not only this group, but also the Rorigula with the Sarracenia, it seems like the ancestral carnivorous plant trap is a sticky trap. Right? We saw it in Pinguicula with Gillicia and Utricularia as well. The one at the base is Pinguicula. So we're not too sure how you get from a flypaper trap to a pitcher trap, but a snap trap maybe you can imagine a little bit more. At least it starts folding on slowly at first in half, and then it goes a little bit faster or whatever. But to Nepenthes, I think the only connection we have for pitcher traps is maybe Gillicia, right? So we have a flypaper trap like Pinguicula and Utricularia. That's sort of like a fast-moving pitcher if you think of it. And then you have Gillicia, which is more of a hollow tube where the prey will go in and can't get out. Okay, it's sort of an underwater or underground pitcher trap if you think of it. Okay, so that's what we saw with these phylogenetic trees, the initial phylogenetic trees of carnivorous plant families, that most groups seem to have flypaper trap at the base somewhere, that then derived into pitcher traps or other types of traps, snap traps. And in this group over here, we see that not only did we develop different types of traps, but we even lost carnivory in a few groups. It seems like they were all carnivorous originally, but then we have Triphyphilum in here. We have some of its sister branches here, Habropetalum, Dancophilum, and Cistrocladus, that are not carnivorous. And Triphyphilum is barely carnivorous. Some of you may know that it has 3 different phases of its life, and it's only carnivorous in one phase. So that's what we learned from the initial phylogenies of carnivorous plants. First, they're not monophyletic, they've had multiple origins. And second, it seems like flypaper traps are the original ones. Woohoo, go Sundews. Okay? So, Nepenthes. I'm going to start with this one just to get it out of the way, because many of you know I'm not a huge fan of Nepenthes, so let's just get this one out of the way. Also because I'm really surprised how many people like Nepenthes, and yet this is probably the genus that's gotten the worst phylogenies. Not because of the author, I'm not blaming the authors, I'm just saying it's a little bit of bad luck of which genes were chosen, as I'll show you. So what do we see here? Polytomies. All over we have these branches that are not well resolved. We have all these species clustered together, and they'll say, oh look, Aristolochioides is maybe closer to this one, but you can't really tell. There's some numbers here that I'm not going to go into, but usually the higher the number, the better chance that that is correct. And I'm not going to go into these numbers because it's been 20 years since I worked with phylogenies. I was a little lazy to go back and study what they were. But anyway, the important thing for you guys is that the higher the numbers, usually the better. But what we see here is that they're all crashing into polytomies, and we can't really tell which species are more closely related. We can see that, yeah, we have some basal ones that, you know, the more primitive, as we call them, the Distillatoria, Pergoli, Metacariensis, Cassiana. But then once you get to like Borneo and Sumatra, it's been a recent evolution. They just recently diverged into species. And at least using this one gene, chloroplast, we can't tell anything. So what do we get from some of these initial phylogenies? That, yeah, okay, we can see some of the basal ones, but once you get into the thick of things here, yeah, you can't really say what's close to what. Furthermore, there was a study published showing that that specific gene that was used, it's a mess. With Nepenthes, maybe it's not trustworthy. There's multiple copies of that gene, and there's sometimes when you extract the DNA and you try to sequence that DNA, you think you're sequencing one copy, but you may be sequencing that other copy, and sometimes you're sequencing both, and that just messes things up. So maybe that's why we're seeing low resolution here. We don't really know. So my conclusion from this paper is that this specific gene, TRNK, don't use it for Nepenthes. But here's another phylogeny that was done years ago. Unfortunately, this one was never published, but it is available. It's a PhD thesis. Yeah, and you can see why it wasn't published, right? I mean, tons of these. And I'm sure he would have cleaned it up a little bit if he was going to publish and take away some of these arrows. But there's something really interesting here. He's comparing a chloroplast gene with a nuclear gene. And yeah, we have tons of polytomies where we can't say that Bongso is more closely related to Spectabilis or Dubia. No, we don't know. It's a huge polytomy. We didn't get enough indels or SNPs to be able to separate them. But there is something really interesting. I think there's a huge lesson learned in this study. Look at this. If you compare one tree to the other, nuclear chloroplast, look at Alba marginata. In one tree, they're showing up together. In the other tree, they're showing up apart. And that happened multiple times. Look at maxima. I have two maximas here and way separate there. Why is that happening? And this is what I want to start getting into. Why some of you have looked at phylogeny and said, this is crap. This doesn't make any sense at all.
Maybe they're all different species.
Yes, that's probably all those different species. Oh, jeez. But even so, why would two different species be lumped here and separated there? Hybridization. We have tons of hybridization in Nepenthes. It's been going on for millions of years, especially with these lowland guys where they all mix up. And especially when, you know, humans come along and build roads that connect what used to be separate habitats, and you start growing along the roadside. Anybody who's been Southeast Asia, you'll see Nepenthes by the roadsides. So what's happening here? We're comparing, remember, a chloroplast gene and a nuclear gene, so they show different things. You see the chloroplast gene here shows them separate. So what does that mean? That maybe there's an ancient hybridization event, who knows, maybe 10,000 years ago, maybe 1,000 years ago, maybe 100 years ago, where two species mixed, but then there was introgression, right? That hybrid kept back-crossing with the parent, back-crossing with the parent, but that little chloroplast from the ancient hybridization event is still there. Who knows, maybe that, let's see, who's close? Berkei, who knows, maybe that chloroplast is actually from Berkei or some other extinct species that we don't even know of. So when you start seeing different things like that, when you compare nuclear trees with chloroplast trees, you say, introgression, hybridization, and think Sarracenia here as well as I'm saying this. So, again, depending on which gene you choose, whether it's from the nucleus, from the chloroplast, you may see completely different pictures. They may be biased. Some genes work for some families, some don't. And to be honest, I really haven't seen a phylogenetic tree for Nepenthes yet that isn't tremendously biased and full of polytomies. I think there's a lot more work that needs to be done with Nepenthes. But let's forget Nepenthes. Let's go to the important ones. Sundews. Not just the sundews, but the whole family. Genus Flytraps, Aldrovanda. Again, you got different trap types in the same family, right? And a big question that we had, were the snap traps, did they have a single origin? Or did it evolve twice? Did Aldrovanda and genus Flytraps maybe evolve from completely different branches of ancient sundew-like plants? Well, here's an example where they sequenced 4 different genes. One of them is nuclear, 3 of them chloroplast, and we got completely different pictures. So we know already that Drosera regia from South Africa is the most primitive, right? The one that's at the base of all the sundews. But look at that. Sometimes it shows up at the base, but we got a little polytomy there. Which one is more base, Aldrovanda or Dionea? Sometimes we get regia showing up with Aldrovanda and Dionea, supporting what some people have proposed, that maybe regia is a separate genus of its own. So looking at these 4 genes alone, who knows? Maybe we should put them together. And that's what's done in a lot of phylogenetic trees. They'll get the sequence from multiple genes and put them together. Sometimes that makes it better because you're putting more characters in there. Sometimes it just messes things up even more. In this case, it seems to make a little bit more sense. Seems to agree more with our bias that regia is really a sundew at the base of all other sundews, and that Dionea and Aldrovanda are more closely related to each other. Maybe there's a single evolutionary origin. And they use 4 different genes. You think, oh, that's much better than the other one we saw, which was only 1 gene or 2 genes. But adding more genes doesn't always help. So again, I'm biased. This is my paper from years ago. This is my master's work from 20 years ago. I did a small tree where I sequenced 2 different genes, RBCL from the chloroplast, 18S from the nucleus, and their combined sequences actually looked better than the previous one with 4 genes. Why? Because I was accidentally lucky to sequence Arturia and discover that Arturia is the second most basal sundew. And so that's sort of, when you add more species, sometimes one branch tends to pull the other one, attract the other one, and give you a more clear picture of what's in there. So adding more species actually helps phylogenetic trees a lot of times. So in this case, merging these 2 genes, we saw, okay, this is more what we expected. That makes a little bit more sense. But back then, it was still a lot of work to sequence. Man, I spent 3 years trying to get all these species over here just for 1 single gene. It was a lot of work. And I was able to get a lot of sundews from across the spectrum, from across the whole genus, all different sections, almost all sections. But look at that. Looking at a single gene, for me it showed up that Aldrovanda was most closely related to regia. But the important thing that I was able to show, that Arturia is the next one up on the tree, that we have a big Australian group of species here where the Phylogenetic complex is close to the pygmy sundews, which sort of makes sense if you look at the plants, right? A Phylogenetic complex is like a big pygmy sundew without gemmae. And you have here the tuberous Drosera close to Banana. That was a bit of a surprise. Hey, the tuberous Drosera, Banana, I don't know, fleshy roots, who knows? Anyway, that was something pretty interesting. And I didn't get too many species in here, so maybe once we get more sundews in there, we'll see bigger groups. The flat rosetted ones with the long-stemmed ones with the stolonifera separating into different groups. Maybe not, who knows? Pygmies as well, there's a lot of space in there. And after a few years later, we sequenced that Drosera myristocalis from South America, and it landed smack in the middle of the pygmy sundew, showing that Drosera myristocalis is the pygmy sundew that somehow made its way to South America and lost its gemmae. We got Glandolidra there at the base of the Australian branch. Many of you know Glandolidra is a little annual species. It looks a lot like Drosera bermanii. And look at bermanii, right over here, close to Sicilifolia from South America. We knew they were close. They're almost identical, indistinguishable. Somehow Sicilifolia seems to have been a bermanii that made its way to South America, just like myristocalis. And then we got a really weird grouping here that was totally unexpected. We got the Adelaide group, Prolifera schizandra, close to a branch with the indica complex and Drosera hamiltonii. Mostly Australian species, so we got Australian species here, mostly Australian species here at the base as well. And then we get into a branch with the non-Australian species. Everything else, Africa, Europe, South America, North America. Starting with Uniflora from Chile, Argentina, as well as Stenopetala from New Zealand. Right? How can a plant from New Zealand be closely related to one from Argentina, Chile? Maybe it migrated through Antarctica at some point when the world was warmer. And then we got a really interesting branching over here. We got basically three groups that were pretty much unexpected as well. One group of mostly North American New World species, or circumborea, that is the northern temperate bogs, mostly diploid, 20 chromosome species. Then we got a branch of Brazilian species, tetraploids. Then we got another branch of African species. So sort of divided by geography, by the ploidy level. But you know what? Polytomies. We got lots of polytomies. So we're able to see that there were different groups, but how species are related in there. This seems to be more a recent divergence, so we weren't with a single gene. Where is it, RBCL? I wasn't able to get too much resolution in there, but you know what? I was extremely freaking lucky. I chose one gene and I just went with it, and it just happened that that gene actually worked pretty well to resolve the bigger picture in sundews. I could have gotten a gene where it would have shown me polytomies throughout the tree and I wouldn't have seen anything. And like I said, it was 3 years of work, so I was extremely lucky in choosing that gene. But what I'd started out with, of course, me being from Brazil, I really wanted to solve that group of Brazilian species and be able to have more evidence to show that, Graminifolia was different from Spiralis or whatever. So, in the end, I was happy, but that little group over there frustrated me a lot. So I was thinking, if I ever go back and do this again, or if somebody does this again, I'd really like to dive in deeper in this group over here and try to solve the different branches here as well. So we thought, hey, how do you solve this thing? Just add more genes, right? Sequence more genes in there, and that'll increase the resolution. That's the way the phylogenists usually think. Well, somebody just did that. So unfortunately, Andreas left. Andreas had a PhD student from Brazil, Paulo Donella. Some of you may know him. He's a carnivorous plant guy from Brazil. I've been in the field tons with him. He added one more gene, ITS, nuclear gene. So remember how I said that ITS is nuclear gene, RBCL is chloroplast. Often when you merge sequences from different genes and you add it all up, sometimes the tree becomes better. Well, in this case, not really. You have tons of polytomies in there. Luckily, they did a few more genes as well, which when they publish, they'll add those other genes. Maybe it'll help clear things up a little bit. But we got tons of polytomies with things all sort of mixed up in there. So adding another gene, unfortunately in this case, did not help us understand the relationships within sundews any better. And furthermore, this ITS, remember how that other gene in Nepenthes didn't work too well, the tRNA and tRNK? ITS seems to be crap in sundews. We're not sure why. We're not sure it's because, remember, it's that gene that has multiple copies, and are we sequencing the right one? If Andreas was here, he would tell you need to clone the genes before you sequence. I won't get into what that is, but what I get from this is that I would not trust ITS too much in sundew phylogenies. There's just something happening there, and there has been a paper published explaining that, these genes are complicated. They may have multiple copies, and then yes, you might have to clone them before you sequence them, whatever. So 18S, ITS, if anybody in the future plans to work with sundews and do, a master's PhD, avoid this gene, okay? And here's another work that was done that I'm going to leave this one anonymous because I feel bad for them. This is a PhD thesis that was never published. It's from 2006. And here we go, ITS. Phylogeny done completely with ITS. And you know what? We got some crazy, I won't say there's kids in here, crazy stuff in this tree. We got some Pediolaris in here mixed with Hamiltonii, with tuberous Drosera. It's like, what's happening here? This doesn't make any sense. And then you got over here some other ones. Wait a second, you got South African with Brazilian, with the diploid Brazilian. Wait, no. This is just not making sense. But hey, it's ITS. And like I said, I don't trust ITS, at least for sundews. And here again, look, Capillus with Adelaide, with Schizandra, with Harmariorum, Versali. Any of you who knows, who cultivates carnivorous plants think, hey, these are not related. There's something went wrong here? Unfortunately, their other tree, the mat-K, which is chloroplast, which tends to work a little bit better for sundews, this one is also really weird. We got, again, look at this. Pygmy sundews, Adelaide, Schizandra, Communis from Brazil, Madagascariensis from Africa. Like, whoa, wait a second, no. Even this gene seems to be a little odd in this phylogeny. And the same thing over here as well. So my conclusion from this work, and mine keeping it anonymous, I think there was contamination. You know, in the lab, you're sequencing this stuff over and over. We're using a technique called PCR, which grabs one little snippet of DNA, multiplies it millions of times, and sometimes these little bits of DNA get around the lab. I had a similar issue when I was doing my phylogenetic tree, when I presented at the Bonn conference, and ICPS conference, what was it, 97, 98. I was showing a Brazilian species, Drosophila I was branching completely separate, closer to Australian species, and afterwards, looking through my data, I realized that I'd accidentally switched one of the files and had mixed Strachii with, I think it was Pediolaris. Like, oops, correct that. Luckily I caught it before I published it, but you know, sometimes contaminations happen. They're very common in labs, and you need to go back and resequence and reconfirm. So here it's not only a factor of weird genes acting in weird ways, but I think also there must have been some mix-ups either in samples or contamination or in the actual data files, and maybe this is why this was never published. But I just want to show you guys that things can go wrong, and a lot of times they do. Okay, so a conclusion, every plant family has good genes and bad genes. Some of them work, some of them don't. Some of them work for one family, some of them work for other families. And bad things can happen. I mean, contaminations, switching files, and just bad luck. And again, I was extremely lucky that I chose one gene that worked relatively well. So here's a case where things went wrong. But maybe not because it wasn't the scientist's fault. I think it's Aldrovandus' fault. So Aldrovandus, some of you know, is an aquatic species that is distributed throughout the old world. You'll find it in Europe, in Asia, in Australia, in Africa, and they sequence 3,700 bases, 3,700 A's, T's, and G's, which is a lot, actually, comparing to other phylogenies. And they use another method called RFLP as well to compare. And they got almost absolutely no differences at all between 15 different plants from all over the range of the species, showing that it's extremely uniform. This is suggesting that Aldrovanda maybe went through a bottleneck a few thousand years ago, and maybe it was reduced to a single bog, and then from there it expanded again really quickly, like aquatic Utricularia often do as well. Who knows, maybe aquatic birds carrying them around. But it shows that Aldrovanda nearly went extinct in the very recent past. So there was a genetic bottleneck that makes them all look alike if you look at the gel. Of course, this is looking at, 3,700 bases of chloroplast gene. Once we look at the whole genome, maybe we'll find more variability in there, who knows. But this initial study, yeah, we can't tell where Aldrovanda originated. Did it start in Europe? Did it start in Australia? Who knows? Moving on, Phylcoxia. And I added some pictures here just because most people don't know what Phylcoxia looks like. It's the most recent carnivorous plant genus. It was only published in the year 2000 with 3 species, and currently I think there's 6 or 7 species. They are extremely rare, isolated to sand dunes, a single sand dune sometimes. They grow in very sandy habitats, and they have little glandular leaves that are sitting close to the surface, often covered in sand, and they trap nematodes. Each species is only known from a single population, except, let's see, one of them has 3 populations. We discovered 2 a few years ago. Another species, I think, has 2 populations, and another one has 2 populations. All the ones are a single population. They're extremely rare. So nothing too interesting here. Most of you don't even know the names of the species, but I just want to show where Phylcoxia is located. For those of you who like non-carnivorous plants, you may know what those plants are. I don't. So they use a lot of different genes, both nuclear and chloroplast, to compare and see where they're located. Plantagenaceae family. And then this is not published yet. This is from a PhD thesis. It will be published soon with some of the species. I think they added a few more. And you have 2 species from northeastern Brazil, 2 species from central Brazil. So maybe a geographic distribution there, but I just want to show you that phylogenies are being made with all different carnivorous plant families, including the most recent one, Phylcoxia. Biblis. Some of you may know Biblis. There's 2 perennial species, and I don't even know how many annual species there are now. Maybe 5 or 6. Who knows? So So somebody did a little phylogenetic tree there with Biblis, comparing a nuclear gene with a chloroplast gene. You know what? They sort of matched. It pretty much shows, perennial species on one side, annual species on the other, with a little grouping here of Phylofolia close to Aquatica, Gehoii close to Lindiflora, Orida on a separate branch. So it seems like this worked pretty well. And it did some other genes, RBCL, TRNK, adding them both together. Gigantea showing up at the base, so maybe they're originally all perennials and then it became annuals. Who knows? Okay, so again, just to show you that this is also being done with Biblis, phylogenies are being done. I imagine we're going to see more in the future, maybe more genes, more data, more species, more individuals per species. Let's talk about the lengths, starting with my favorite, Gillicia. So, and again, I'm biased, I'm over here. But this came out really good. I'm really happy with this. When we sequenced the Gillicia, we saw, hey, we got what we expected. All those purple Brazilian species clumping together in a single branch, showing, okay, they're monophyletic, they have a single origin. You got Ensenada, Violacea, Lobata, and over here we haven't yet described these species, but it's Flexuosa, Oligophylla, and which one? I think that's it in there. Which one? We didn't sequence that one, unfortunately, but yeah, it would probably show up in here somewhere. And then we sequenced some African ones and some other South American ones, and we found something really interesting, actually, that it seems like they originated somewhere where Brazil is currently, or South America, then somehow they jumped over to Africa, several species evolved there, and then somehow they jumped back to South America, and the continents were already separated by then, so yeah, they did jump the Atlantic, however small or large it was, but it's just interesting, Gillicia went to Africa and back. And we got, so 3 main groups there, the basal, Brazilian, purple-flowered ones, and we got the African ones, and then we got the mostly yellow species from South America, with a few exceptions. At the base, we have Guianensis, Sanariapoana, which are purplish, more similar to the African ones, and we got Aurea, Repens, Roramensis, etc. from. So this actually matched what we were expecting. It actually gave us a few new clues of different groups that are in there, which we named afterwards. Now I don't remember the names. You have to ask Andreas or read the publication. But this phylogeny actually worked pretty well. We only use a few genes there, not too many, and these are all chloroplast genes. So again, once we look at nuclear genes, things may change a little bit, but so far so good. Next, Pinguicula. Here it's a little bit messier. There have been a few phylogenies, and we got a lot of polytomy, especially with the European ones. It seems like they diverged relatively recently, so we can't really tell who's closer to who. But hey, we got some interesting views here. The Crystallina hirtiflora group from the Mediterranean seem to be basal to all Pinguicula. And we got a little branch there with the North American species, maybe being the next basal. But this is only nuclear genes, ITS. Looking at chloroplast genes, you know what? It doesn't look too different. We got a little polytomy here with the Europeans. We got, again, Crystallina at the bottom. So how do the two compare? So we got ITS on the left, nuclear, chloroplast on the right. You know what? They look pretty similar. We got Crystallina hirtiflora at the base. Next branch up, you got the North American species, and we even have the South American species in here, Antarctica. A lot of the South American species do look like the ones in the southeast. If you think of planifolia, for example, right, the red leaves, the purple flowers. If you've ever seen pictures of Pinguicula calitrata or involuta from South America, they look very different. They're just smaller than planifolia, but they're very similar. Did I say different? Yeah, they're very similar. So, what else? Mexican Pinguicula. So, in this branch, Mexican Pinguicula were next up, and this one is further up the tree. Actually, if you look here, there's a European branch, which I think, yeah, the European and the Mexicans. In trees, you can flip the branches. It doesn't mean that this branch over here is closer to these guys. You can flip this equally, and it doesn't really mean anything. So what I'm showing you here, it's not that European or most closely related to North American. It could be either way. So the trees are really matching. I mean, you could flip this branch and it would look almost exactly like that one, where you have some of those Asian species there, a European, Ramosa, Velosa, Variegata. Sorry. These are all grouping together on both trees, sort of in between the European and the Mexican group. So yeah, that's making sense. The only thing that was weird in this tree, Lusitanica. In this tree, it's branching up right at the base near Crystallina and Hertiflora, whereas in this tree it's sort of in between the North American and the Mexican. So, not too sure. Otherwise though, surprisingly good. Oh, and Cuban species here, Phylogeny. It seems like the Cuban species are most closely related to the Mexican species, which makes sense. Whether the Cuban species are monophyletic or not, we're not really sure yet. So here's a phylogeny that was published in the Mexican species. Yep. A lot of them, a lot of polytomies, we're not too sure yet which are closer to which. I think we need a little bit more resolution because only a single gene was used here. And here's a phylogeny of the Cuban species where it looks like they're monophyletic. And it's the same authors, but they didn't merge the two trees. And rumor says that when you do merge the data from the two trees, it all becomes one big polytomy. But that's why they publish it separately, allegedly. I don't know. I didn't see the data, but we're not too sure yet then whether the Cuban species had a single origin from the Mexican, the one species that jumped over from Central America maybe, or whether there are multiple origins. That's still being debated. This is one of the most recent papers with Thingwick, and I want to show this one because it's a trend. We're going to start seeing this more and more. They sequenced a whole bunch of European species, and we see tons of polytomies in there. You think, crap, it didn't help anything. But wait a second. We got a new species out of this paper. They sequenced a whole bunch of penguin, and they saw that there was one group of species, of plants that were clumping together, separated from everybody else, with really high resolution here. This was a previously undescribed species. Yeah, I forget which one was classified under, Vulgaris, Grandiflora, whoever. But these noticed, hey, this population here, this area, seems to have its own little monophyletic group, and they named it. It was based on a single gene, so maybe it was a little preliminary, but let's say it helps feed further studies, this sort of thing. Cetebensis. It's from Spain, I think. Yeah, I think it was Spain. Anyways, there's a paper. I can share copies of the presentation if anybody wants it afterwards. I put all the references there so you guys can go look them up if you want. But basically what I want to say with this is that we're going to start seeing this more and more. Traditionally, taxonomists go in the field, they collect tons of plants, they make herbarium specimens, they collect DNA leads for DNA extraction, then they sequence, then they look into it like, oh, look, this is surprising, this is surprising. Let's go back in the field and look at these plants. Maybe there's a new species there. Yeah?
Did they confirm that crypto species with morphological features after the fact, or did they do it simply solely on molecular data?
There are characters that place this one apart, and sometimes it takes molecular, and that's what I was getting with my point right now. So this is really interesting, where traditionally botanists have gone in the field, collected things first, then sequenced, and then gone back in the field to look again, are there characters that put these guys together? And in this case, there were some characters that they described, say, hey, it does look like these guys deserve to be a new species, not just based on the DNA sequence. But more and more, I think we're going to start seeing the opposite. Field work is a lot of work, right? You have travel expenses, you have to climb mountains and face all sorts of hazards, mosquitoes, whatever. What is beginning to happen, and what I think we're going to start seeing more and more, is that we're going to start sequencing first, figuring out what is what is branching with what, and then looking, look, this branch seems like it deserves more study. Let's focus our resources on going in the field and looking at this group, instead of just going haphazard, collecting anything and everything, and then trying to make sense of the data. Botanists tend to be very conservative, and it takes them a long time to adapt. A lot of them are afraid of doing lab work, but I do think we're going to start seeing this more and more. And this is a clear example of this, okay? So this is, I think, one of the first examples that we've seen in carnivorous plants. Utricularia. There's been a lot of work done with Utricularia. It's actually one of the most interesting genera in terms of phylogenies. So starting back in 2005, we had this phylogeny that was published using 2 chloroplast genes, and you know what? It's pretty much matched what Taylor had said in his monograph of who was closely related to who. We were very happy to see this. Hey, this is a case where the morphology of the plants is matching the phylogeny, the molecular phylogeny. So, okay, had some interesting groups in there. Nothing too surprising. Maybe subulata and that group being close to some of the aquatic ones is a little surprising for me, but otherwise, pretty much what we expected. You got multifida, but the polypompholics at the base, more primitive. Some of you may have heard that lately polypompholic, the old polypompholics, which are only 3 species, multifida, tenella, and westonii, have been found to have passive traps. right? All Utricularia have active traps that suck the prey into their traps. The old polypompholic species of 3 have passive traps. They're more like a miniaturized Gelicia trap than a Utricularia trap. So that's something really cool. They're showing up pretty much their base, showing sort of an intermediate step. Yes, Bill, again.
The size of the overall genome of these.
Oh, I'm going to get there. You're good. He's catching on where I'm going. So then there's more phylogenies. This one was from, when? Recent one, 2018. So they added a lot more genes, a lot more species, and nothing really changed compared to the previous ones. A few little details here and there. And there has been some discussion of, where is it? Oh, I think it's in the next slide. Yeah, so I couldn't fit the whole tree on one slide. I had to split this one. So there's been some discussion regarding, where are they? There we go. The group called Eperua and Orchidioides, where some are saying that they should be merged, but I'm going to show you why this is being said. Oh, and a little disclaimer here. Most of you have probably never heard of this species. It's a really rare species from Brazil. I thought I discovered it many years ago, and I sent material out to people so they could include in phylogenies, and that was a mistake. This is actually a new species in the Amethystina tricolored group. I only noticed it later. So unfortunately, people are still using this sequence that's in databases on the internet and calling it Huntii, and I'm sorry, it's not, okay? So this guy actually belongs in this group over here. It looks like a Calliciphyta, actually. But, so wherever you see this, sorry, my fault. No, it's not, because it's so rare. Nobody has it, right, Damon?
Yeah.
Hopefully that guy will spread it to everybody. Okay, so there's the only big discussion that there is right now that I can see, and maybe I'm biased because it's the Brazilian South American group, is this Orchidioides eperua group, where they've sequenced MATK, chloroplast, and ITS nuclear, and they're not matching. And this one is showing that Humboldtii is closer to the Orchidioides group, when we actually thought it was closer to the Reniformis and Nilbifolia group. And this one's really weird. Carnivorous, which is close to Reniformis, and Nilbifolia showing up in the middle of the Orchidioides. So I don't know if ITS is the best gene for Utricularia based on this alone, but based on this alone, people are saying that they should merge these two, and again, that's only a single gene. We've seen that when you sequence different genes, even different genes within the chloroplast, you see different trees. So you know what? I would hesitate before merging these two based on a single gene. That's the only really weird thing that I've seen in Utricularia. But here's a cool new paper that came out recently. This is from 2017. Richard Jobson in Australia has been very active with Utricularia, publishing tons of new species, and he did something amazing. He did 205 accessions. What are accessions? Different samplings from different species. He did a huge phylogenetic tree, and that's only a part of it. This is the other half. He used 3 different genes. He sequenced tons of plants. And when I say tons of plants, I mean not just tons of species, but a lot of individuals per species from different populations from across the range. This is what I hope to see with a lot of carnivorous plants in the future, where you're not just doing one Aleda, you're doing a whole bunch of Aledas from the east side, from the west side, and how are they related to each other? And he got some really good results where every single species was basically clumping together with the other individuals of its own species, showing that, yeah, okay, most of these species are monophyletic. Of course, there were some complicated ones, so you're going to have to wait. Let me finish this sentence here. Well, we expected to see a lot of problems. Dichotoma, Monanthos, Novazelandiae, we know these guys are closely related. There's been a lot of back and forth. Are they different species? Are they a single species? Are there multiple species? So it's not surprising that we saw polytomies in here. And, you know, we're going to need more genes or more sequence, whatever, to try to figure those guys out. Is there really hybridization going on here? But really cool that, again, he sequenced some unpublished species. And they pretty much all showed up as monophyletic, showing that, hey, these are individual little branches here that probably do deserve to be classified as their own species. And he recently published, I think, 6 of these. I think it was 5 or 6 new species from Australia. He's been publishing tons of new species from Australia. But that was a really cool thing, again, where he collected tons of stuff. He sequenced them and he saw, yeah, you know, what I'd seen in the field, and maybe there's some morphological differences. It's confirming here. You're adding more data. You're saying, yeah, there are different species that probably need to be published. Yes, Bill?
So those last names, the numbers, those are collection numbers, and those are actual?
Yes. Sorry.
And why is that important?
So these are basically herbarium specimens that were collected. So Richard Jobson, number whatever, and I think this is probably how they can put it in the herbarium, but either way, these are different collection numbers. So when you go in the field, you make a herbarium specimen, you give it a number so that you can reference it later and say, hey, the one I'm sequencing is that particular herbarium sheet that's sitting at whatever institute, and it has that number. Yes?
Are you actually taking samples from dried herbarium samples, or do they store those samples in another way?
So good question. Getting good quality DNA is extremely important, and sometimes herbarium specimens give cracked DNA depending on how quickly they were dried and which organ you're using. So Andreas, for example, will always tell you that if you get DNA from flowers of the lentibullariaceae, you get better quality than if you use leaves. In this case, I know them, so I know that when they're in the field, they're not only making herbarium specimens, they're also collecting fresh leaves and putting them in what we call silica gel, which dries the leaves really quickly and preserves the DNA. Yeah. So when you know what you're going to do with that material, and you in advance, and you take this silica gel and you dry the leaves, you can get pretty good results. There's a problem with old herbarium specimens or material that didn't dry too quickly. The DNA degrades, it gets fragmented, and it gets a lot harder to work with. And this is not a phylogeny necessarily, but I just want to show you again where DNA is taking us in terms of helping us understand the relationships between these species. This is a paper from 2005, where they compared 3 species, actually 2 species, one was considered a variety, so Australis forma tenuicaulis with mycorrhiza, and they compared several chloroplast genes as well as another method that I won't go into called the FLP, and they found that Australis is not only sterile, but it's a hybrid between tenuicaulis and mycorrhiza. And it wasn't a hybrid that just happened once, it's a hybrid that keeps happening over and over again, sometimes with mycorrhiza as the father, sometimes with mycorrhiza as the mother plant. And yet Australis is probably the most common, it is more common than tenuicaulis. It's spread further just because it's an aquatic, it's carried by birds, whatever. But we're going to start seeing this more and more as well, where they're comparing different species and seeing, hey, this one has a hybrid origin with these guys. In some cases, they may be fertile, sometimes may be sterile, sometimes it's hybrids that are still happening, sometimes it's ancient hybridization. But this is the power of DNA. This is what it can show us. And finally, Sarracenia. I left this one for last, the Sarraceniaceae, because this is where I think the best studies have been done in terms of phylogeny. So I know a lot of you like pitcher plants, Sarracenia, so lucky for you, there have been some really interesting studies. So as we saw from the beginning, the big surprise with Sarraceniaceae was that they were close to Origula. So again, we had a flypaper trap sort of basal to the pitcher traps, showing that maybe originally the ancestors all had flypaper traps and then somehow evolved pitcher traps. But the first phylogenies we saw with Sarracenia, oh my gosh, this doesn't make any sense. And Polytomes, no support for the trees, okay. And the big surprise as well, Darlingtonia at the base, followed by Heliophyra and Sarracenia, but I don't know, is Leucophylla really closest to Purpurea? Is Psittacina closest to Flava? There was only one collection for each species, and who knows whether they were pure or where they were from. So then there was another one in 2012. They used more genes, right? Remember I said phylogenies, if one gene doesn't work, add another gene, add another one. Keep adding genes, and hopefully we'll get better. So these, they combined a whole bunch of different genes, I forget, but I think some of these, we have nuclear genes, we have mitochondrial genes, we have chloroplast genes, and you know what? It only seems like it got worse. Okay, we got the Purpureas here on one little branch, but not too much support. But then, geez, we got a Polynome here where we got Aureophila with the rubras, with the minor, Aleta. Okay, again, this didn't really help. But then there was another paper that was really interesting that it didn't really help the phylogeny either, but it just shows a little bit of what we expect, which is the gene flow. So they use a method called microsatellites, which I'm not going to go into, but basically showing that there is gene flow between different populations, between different species, and sometimes one species contributes disproportionately, maybe because it's just more abundant, maybe because it flowers not in a specific month, but it flowers more often and it has more chance of hybridizing with others. Who knows? But the important thing is, the important message that I got out of this paper is there's gene flow. And if there's gene flow, when you look at single gene phylogenies or few gene phylogenies, especially those that are from chloroplasts, you're going to see some crazy stuff. It's not going to necessarily make sense. Which is where this paper comes up. So this is from, what year is this? 2012. So they use a really interesting method. I don't necessarily agree with the conclusions of this paper, so let's ignore what they concluded. I know there's some contention there, some of you may know this. Let's ignore the conclusions. The important thing that I want to focus on here is the method that they used. So instead of using a single gene phylogeny, 2, 3 genes, they use something called reduced representation libraries, where you're basically sampling a subset of the whole genome. You're grabbing like 1%, 2%, 5% of that whole genome and sequencing it across multiple species or multiple populations, and then comparing that. So you're getting a lot more data than a handful of genes. And they actually found over 500 SNPs, which is, believe it or not, a lot to compare these and do some sort of phylogenetic work. And again, not necessarily agree, because they're sort of proposing that there are maybe 2 different species of Elida, one west of the Mississippi, one east of the Mississippi. Again, forget the conclusions. The important thing is that this is the first study of carnivorous plants where they use what we call next generation sequencing, where they gathered a lot more data to compare, not just a handful of genes. So I think this is important just because it's a trend. We're going to start seeing this more and more. So reduced representation libraries, I don't want to go into detail. Again, it's just a sampling of the genome. So we're selecting a small part of the genome and sequencing it from multiple species, multiple individuals, and comparing it and hopefully getting a lot more data. And we're getting a sampling of the total DNA of the genome. So we're getting nuclear genes, chloroplast genes, mitochondrial genes. We're getting a little bit of everything in there. So that's just a way of getting more data. Why didn't they just do the whole genome? Because it was still too expensive. This was, what was it, 2012, I think? It was still too expensive. Nowadays, it's getting cheaper and cheaper. We'll start seeing whole genomes. But here's the paper that I think is most definitive. It's the most recent one as well. This one is from, where's the date? There we go, 2015. Here's one where they used a similar method. They fished out 199 genes out of the pool of DNA for each one of their samples. They used 75 different samples across all the species, all the varieties. So they have several samples from each species, each subspecies of Sarracenia. And they also did a whole chloroplast sequence. Remember, the chloroplast has a little circular DNA, so when you sequence the whole chloroplast instead of a genome, we call it a plastome. So they did the 2 things. They fished out 199 genes from the total pool, and they also did a whole chloroplast. And let's start with the whole chloroplast. Damn. Polytomes, and we see some really crazy stuff. Look at flava. Flava's all over the place. You know, somebody just looked at this and they're going to say, oh, look, maybe there's ancient hybridization. This flava is close to Psittacina. Maybe that's why it has the cutthroat, or who knows, whatever. And then this one, Rubicorpora is close to minor. Maybe that's why it's redder. Again, look, it's full of polytomes. You can't really interpret anything here. Okay, so even sequencing the whole chloroplast, a total of over 42,000 base pairs, it wasn't enough. Why? Too many hybrids in Sarracenia, too much hybrid origin. We know there's gene flow going back and forth. We're not going to solve the phylogenies even with the whole chloroplast sequence. But what if we look at the 199 genes? Well, that's going to get a little bit better. Oh, before I get there, look at that. Rosia as well, all over the place. Psittacina's all over the place.
Wow.
But once we look at the 199 genes, things change. And then we get a much nicer tree. And if you look at these numbers, 100, 93, remember, the higher the number, the better, the more chance that that is correct. And then we start seeing some really interesting groupings and some things that make a little bit more sense. And we know that there's a lot more data backing it. There's a lot more evidence supporting these trees, 199 genes. And we start seeing some interesting things, you know, relationships. Hey, look, all the purpurias are clumping together, you know, whether Montana or Venosa are separate or not. Maybe you need a little bit more data in there to be able to separate these into species or subspecies. We got the minors together. Psittacina's close enough to minor that I'm happy, but then we got the flavas there. Wow, flava close to Psittacina, who would have thought? Arubras are all showing together, and close to Alata, there's a little polytomy there, and you know what? I just went to the field about a month ago, saw a whole bunch of Sarracenia for the first time, and when you see a lot of Ruba without flowers, at least for me, I was confused. I was sending pictures of people who knew this stuff, and they would always ask me, oh, but where is it from? I'm like, wait a second, is your taxonomy based on geography or morphological characters? Right? So basically, a lot of Ruba, if you don't have flowers, they can be hard to tell apart. Huh? Oh, I'm sure Damon could, of course. Of course Damon could.
Do you think that the Elida and Rubra are not definitely because Rubra is like a hybrid origin ancient thing with like Elida and Luke?
So the good thing of looking at 199 genes is that if there isn't some ancient hybridization and it's been sort of weeded out, introgressed back in the parent, that little bit that might still be in there, with 199 genes, it tends to be weeded out even further. So this, I think, shows a bit of a clearer picture of where these species are located. Of course, we need to look into it further, maybe do whole genomes to find out, what percentage of that introgression is still in there. But it's much less biased, let's say, than a handful of genes, chloroplast genes, which would only show maternal lineage. Okay? But there's one interesting thing here. For that whole discussion, is Alabamensis a good species or not? Well, according to this tree, even though there's a little polygamy there, Alabamensis seems to be more distantly related to the other rubras. And not only that, but some people say that Warei is in Alabamensis. Well, that's not what this tree is saying. Again, we need more data to be sure, but some interesting trends here that need to be looked into. Okay, so this to me is, out of all the carnivorous plant phylogenies, of all the different genera, all the different families, to me this is the best one so far. And this is what I hope to be seeing in the future with other groups. So what should we expect to see in the future? What are the trends that we're going to start seeing in terms of DNA sequencing being applied to our carnivorous plants? Well, there's already been a phylogeny published with whole chloroplast genomes, so 151,000 bases, across the Lentiviolaceae. Right? As we saw with Sarracenia, whole chloroplast didn't really work, but here, once we add more species, we add more data, maybe it could help us identify some things. At least it's a good trend, let's say, right? It's not a handful of genes. We're looking at a whole chloroplast sequence there. I want to see more of this. And back in, when was it, 2013, we had the first carnivorous plant genome sequenced, Utricularia gibba. Why? Because it's, who mentioned it, the small genomes. A few years ago, we found out that Utricularia and Gillicia, some of them at least, have tiny, tiny genomes, the smallest known genomes among flowering plants. And I'm going to boast a little bit. The smallest one is actually a new species that I discovered a few years ago, thanks to the help of Thomas Carroll, actually, Gillicia tuberosa, the only Gillicia species with tubers, which he found many, many years ago and was telling me about it. I kept thinking it was crazy for years and years until I found it. But it actually has the smallest it has the smallest known flowering plant genome of all. It's 50, I think it was like 60 megabase, 60 million bases only. Remember, 60 million? We are 3.2 billion humans, right? Anyways, so fantastic. We saw the first full carnivorous plant genome, Utricularia gibba. It's relatively small, only at 80 million bases. Excellent. A few years later, we started seeing more full genomes. We had Cephalotus, Dionea, Sundus, the Joshua capensis, and we're going to start seeing more and more of this. Right? Gillicia has had some interesting papers as well. Gillicia aurea has had its full genome sequenced. There's been a paper published comparing whole chloroplast, so plastome sequences among these South American species. And there's even been a paper comparing 3 whole genomes, Hispidula, Nigrocaulus, and Pygmaea. Again, we're going to start seeing this more and more. And we're already seeing this with other groups. A few years ago, we had a bird phylogeny that was published based on whole genomes. Look at this, a whole huge tree with all the major branches. Whole genomes, entire whole genomes compared. Remember how much it took to sequence one single human genome 15 years ago? $3 billion. We can do each one of these for, depending on the size, less than $1,000 nowadays. So it is a matter of money, and the price of DNA sequencing keeps dropping. It's moving very fast. We're going to keep seeing this price drop and allowing us to do more and more research with more and more species, with more and more DNA. But what do we expect to see then? Let's imagine it's 2018, right? So the next ICPS conference. A lot of us will probably be there. What sort of talks will we see? Well, I think we're going to continue seeing this trend where we're going to see not just a handful of genes for phylogenies. We're going to start seeing whole chloroplasts, maybe that reduced representation library where you're fishing out a bunch of genes, maybe whole genomes. And we're going to start seeing less of the traditional Sanger sequencing, AFLPs, rapids, RFLPs, other older methods where you're only capturing sort of a snapshot. What else? We're going to going to be seeing more and more studies, like ecological studies, comparing populations, and the power of being able to sequence more DNA is going to allow us to look a lot further into, for example, I was talking to somebody about looking at Nepenthes pictures. What are they trapping? Traditionally, you fish out the little insect bodies and try to identify them, you know, the legs, the antenna. What if you just fish everything out and just sequence it? See what shows up, right? So that's what we call metagenomics. And you can do that with any carnivorous plant, you know. Get a swab of Drosera leaves, get a swab of Sarracenia pitchers, Pinguicula leaves, and you can find out what, just a bunch of Utricularia traps and sequence to find out what's in there. Gynlicia trap, what the heck is Gynlicia trap? Maybe as our techniques improve, we can get herbarium specimens of Gynlicia traps and sequence them all and find out what's inside. Okay, so we're going to be seeing more individuals, more DNA being applied, not only taxonomy, but ecological studies. Now let's take a little bit of a leap further into the future. Okay, so let's speculate a little bit. If this trend continues, what do we expect to see maybe, you know, another 12 years? When we all come to ICPS 2030 and Stephen is even older than he already is nowadays, and I'm probably going to have even less hair than I have nowadays. What are we going to see? So, I predict that by then DNA sequencing will be cheap enough that we're going to see on a regular basis things like CITES, border patrols, etc., sequencing DNA regularly to identify what's coming through the borders. You know, they find a package of seeds, they find a package of leaves or stolons. They'll be able to sequence that and find out, it belongs to this species, that species. They're not going to... okay. Second to last slide. So they're going to be able to trace where did the plants come from, which nursery did it come from, which tissue culture lab did it come from. This is going to be more of a standard thing. So hopefully this will help reduce illegal CP trade and poaching by being able to trace and identify all the plants circulating in the trade better. Whereas you guys will be using DNA sequencing, just like you have 23andMe, Ancestry. You're going to be able to mail in a sample of your plants, and they're going to sequence it for you and say which species it is. So, you know, all your plant labels, all these discussions on the internet, is it a hybrid, is it whatever, you know, a lot of this is going to go away. And we're going to look back, remember how much time we used to waste on the internet discussing, is it Rob Campion, is it whatever? So, maybe we're not even going to have labels anymore on pots eventually, and I'll get to that. Okay? What else? Nepenthes, right? Is it male? Is it female? We don't know until it flowers. You have to wait until the flower spike comes up until it starts opening, or maybe you can remove the flower and try to dissect it. We're going to be able to send in plants and identify, is it male or female? As soon as it germinates, it has one little leaf there. You can pluck off that leaf, send it to some sequencing service, and they'll tell you. Hybrid parentage will be easier to fish out. So again, is it a hybrid? Is it not? Is it whether it's a wild plant or whether it's something in cultivation, or maybe you've lost the label, right, Steve, and you don't know what that plant is anymore, and you can just sequence it and know, hey, oh, this is a hybrid between Sarracenia lata and rubra crossed with flava. And not only that, oh wait, oh that's in the next slide. So, so again, all this drama around certain species, we're not going to need to waste time with that anymore. You're going to, again, be able to mail in a sample of your plant, just a dried little leaf, and it'll do it for you and send you back the results. Oh, here it is. So not only are we going to be able to identify hybrids, but even quantify and say, hey, this plant is 60% Aleda, 30% Purpurea, 8% Flava, and 2% Rubra. We're going to be able to get to that point. Maybe not 2030, maybe earlier, maybe a little bit later, but yes, we are going to be able to get there. And again, as it gets cheaper and cheaper, everybody's going to be able to afford this sort of thing. And even your pests, you got some pests on your plant. How many endless discussions have you seen on the internet? It's thrips, no, it's fungus, it's virus, it's whatever. Send in a sample, get it sequenced, and then you'll know what to spray. And we might even get to the point where you know, this is a thrips that's resistant to whatever thing you're spraying. You need to use the other brand. And what we see in CP show and sale. So many of you have seen the glowfish probably in aquarium stores, right? We already have genetically modified fish in the pet trade. We're going to have genetically modified CPs. I mean, who's going to be the first to make a genetically modified Venus flytrap, right? I mean, it's coming. You all know it. You've seen how when I started 20, 30 years ago, we had a single Venus flytrap. You know, that's it. Now you go to California Carnivorous and you see that table with all those dozens of carnivores of Venus flytrap clones, hundreds, and they look really different. And you're thinking, wow, where is this trend going to go? Well, they're going to make glow-in-the-dark ones. They're going to make, I don't know, all sorts of different modifications that we already see in the pet trade. We're already seeing it with fish, right? And maybe they're going to incorporate other genes, resistance to fungus. They'll make a Sarracenia that doesn't rot. You know, it's resistant to whatever fungus attacks it, the virus, whatever. And maybe we're going to see, you know, people inserting genes to make different flower colors that are not currently present in certain genera, certain groups. Or maybe leaves that are longer or shorter, or, you know, maybe a compact little raja. Who knows? They're going to. These sort of modifications are already known in plants and animals. They're already being done. It's just a matter of time before they're applied to CPs. So I think, you know, by 2030, we're going to be seeing a few of these in the CP show and sale. And we're probably going to have a separate prize table for them as well, right? The genetically modified ones. But the important thing is, species identification will become easier. We're going to stop wasting time with all this babble about, you know, what is what, and even in the field we're going to be having, you know, not wasting as much time wondering whether species are real or not. Oh, and plus, who was it? Oh, you had that poster, right? Right, did you guys see that poster, California Carnivorous, with the face recognition sort of software where it identifies plants? We already have that. I forget what it's called, but there is already an app that sort of does that, but of course it's going to get better. And if you go on Facebook and you see the facial recognition that they use there, they recognize pictures from when you were a kid. It says that it's you. The technology's there. It's already there. It's just a matter of training it for our carnivorous plants, and we could have an app that identified every single species that we know of. Right, so that's coming. So plant labels, having that ugly little thing in the pots, why? If you can just go with the software, oh look, this is Pediolaris, this is Ordensis, this is Falconeri, and so on. Right? So we're definitely going to see that. The technology is already here. But what about further on? And this is my last slide, I promise.
You're way over time.
Yes. By 2040, I predict that DNA sequencing will have become much cheaper and miniaturized to the point where people like me who like exploring the field looking for new species, I'm maybe going to have a little portable one that I'm going to be able to take with me and sequence samples on the spot cheaply and already identify plants in the field. Not need to take it back to the lab, extract, blah, 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 etc. This will be tremendously helpful, especially because by 2040, I probably won't be able to walk much in the field, so that'll be very handy if I can. Yeah. But anyways, also, drones. Drones are here already, right? People are already using them for carnivorous plant hunting, like some Nepenthes or Pingricula that are up on high walls and you can't really see them too well. Imagine these drones with the facial recognition software we were talking about, specifically for plants, and that they're self-driving drones, that you can just let them go, sit in a bar, drink a beer, and let your drones go in the field and look for plants for you and photograph whatever they find, maybe even collect samples and sequence them on board, you know, little miniaturized sequencers, or maybe bring it back to you. Technology is basically here already, right? You've got self-driving cars, you've got drones, put that together. DNA sequencing is moving faster and faster. Yeah. So I predict that we're going to see a boom of new species with all this technology, but at the same time, in 20 years' time, I mean, if we look at 20 years ago, how many Sarracenia there were, how many there are now, if you think of not just habitat loss, but climate change, what is going to happen in the future, unfortunately, I think these things are both coming together, where we're going to have huge advances in our capability to study and publish new species, but at the same time, it's going to be a race against time. So we're probably going to see a lot of talks about species that are already extinct and or are nearly extinct. At the same time, a whole bunch of new species on mountains that nobody can climb. You need a helicopter up, send your drones. Whereas the CP show and sale, I think that's going to go even further. Oh, we're not going to just have one gene or two genes incorporated. We're going to have a lot of wacky stuff in there, where we're going to have bizarre traps, bizarre flowers, larger, smaller, different colors, different shapes, adapted plants that are adapted to warmer climates. So in a Sarracenia that doesn't need dormancy, for example. Longer lasting flowers, some leaves that will maybe keep the damn flowers open more time, right, so we can pollinate them properly and see them, not have to carry them to work, right, Robert? And fragrances. You know, we already know some genes for fragrances. What if you insert, you know, have a Pingricula that smells like roses or whatever, And I think somebody's already studying which genes in Nepenthes, determine the sex. I'm sure some of you have heard that there sometimes are hermaphroditic Nepenthes in cultivation that will show up. If we can determine which genes these are and turn them on or insert them, whatever, and have hermaphroditic Nepenthes, that we don't have to, waste time and, send me pollen from here to there, and that's, the Tinder for Nepenthes. We get rid of that. Right? If we can just have Nepenthes free, we can always flower and have pollen and pistils and just, maybe different flower scapes, maybe the same flower scapes, whatever, just solve this problem. Maybe that'll help conservancy even, being able to get more seeds like we were talking about. And my final statement. Yes, species identification will get easier on the one hand, but it's going to get more complicated as well because it's going to bring up this topic of what is a damn species. Because again, plants are hybrids. They're all hybrids. We're hybrids. Remember, I don't know how many of you have seen the studies that we're mixed with Neanderthals and Denisovans. So even we are hybrids. Imagine Saracenia Nepenthes and all the other CPs. There's been tons of hybridization events in the past, some of them more recent, some of them more ancient. We're going to be able to fish out the recent ones and say, hey, this is 30% Aleta, 70% Flava. But we're also going to be able to see more ancient hybridization events of species that are maybe not even present anymore, extinct species. So on the one hand, it will make it easier to identify plants, but on the other hand, it's going to make it a lot harder in terms of what is a species. What are we calling a pure Sarracenia flava, a pure Nepenthes tobica? That whole discussion of pure, I think, is going to have to be reviewed and maybe thrown in garbage. And that's it. Thank you, everybody. Thank you, ICPS. Thank you, the ICPS. And my good friend,